Hello, listeners and learners. Today we are going to be talking about carbohydrates as part of our series on foundational molecules of biology. We're going to start this a lot like we did proteins. First, we'll talk a little bit about monomers, then how they're connected, then how the structure of this molecule serves its function. The monomers of carbohydrates are called monosaccharides. So saccharide means sugar. Probably have heard the term saccharin before. It's the same root. Monosaccharides are named based on the number of carbons that are in the molecule. So a pentose would have 5 carbons and a hexose would have 6 carbons. This ending, ose, means that it's a sugar. So if you see the ending ose, that name is giving you information about that molecule, saying that that molecule is a sugar. So I have here 2 examples of hexoses. We have glucose and fructose, but obviously they look pretty distinct. Here we have a 6-member ring in glucose, and here with fructose we have a 5-member ring. So you can classify your monosaccharide by the number of carbons, and that's your pentose hexose, but you can also further describe them by the number of members of their ring. So a 6-member ring such as glucose or a 5-member ring such as fructose, both of those are hexoses and have 6 carbons in them. The monomers of carbohydrates, these monosaccharides, are joined by glycosidic linkages. And that's just the name for what that bond is. And much like with proteins, as we're making those peptide bonds, this is the result of a condensation reaction, which means that water is removed as that reaction occurs. So referring to this little diagram here and the formation of sucrose, we have this hydrogen that's on our OH, and this hydrogen and oxygen as part of our other OH, our hydroxyl. These both are going to join to form this water molecule, which then leaves this new bond with these members, and that creates our glycosidic linkage. Personally, I wouldn't be too concerned about the organic chemistry reaction mechanics of this, because when it comes to the function of sugars in the body, that's not quite as pivotal to understand, but understanding that the construction of this bond requires input energy because you are creating a larger and more complex molecule, that's quite important. Finally, we're going super quick through our carbohydrates. I think it's interesting to observe the range of function that you see in carbohydrates. Carbohydrates are very good for energy storage, but you will also see them used in a structural role. So here I have 3 different examples of carbohydrates. Here I have 3 different examples of carbohydrates. I have starch, glycogen, and cellulose. So these are all good terms to know and have a little bit of an understanding of. So starches are a family of polysaccharides. It's an overall group. There's many different kinds of starches. Here in my pictures I have amylose and amylopectin, which are 2 different starches. Both of these are found in plants. And in plants, these starches are the primary energy storage molecule. So potatoes, for example, that's going to be a carbohydrate sink, a sugar sink. A lot of energy is stored in the potato itself. Glycogen is very similar to starches in that it is a glucose polymer, and it's primarily used for energy storage, but you see it in animals, and this is energy storage in the liver. Then we have another glucose polymer, in this case cellulose, and it's a little bit different than the other 2 in that cellulose does serve a structural function. The other 2 are both used for energy storage. So here we have 3 different examples of complex carbohydrates. So taking a look at their structures, we can pick up on a few cues that would indicate why these molecules would serve those functions. Both starch and glycogen look very similar to each other. They both have this very branching structure, very tree-like veins in a leaf. Now if you think about the way that you're going to get energy out of a complex carbohydrate, you're going to be breaking those glycosidic linkages. You're going to be breaking the bonds in between those monomers, and that is what is going to release energy. If I have something that is this large webbed pattern, there are a lot more ways where if all at once I wanted to start breaking apart linkages, I could start here and here, all of these different sites. Glycogen, very, very similar structure. It would be the same sort of approach. There's a lot of different points where you could start breaking apart linkages. Cellulose, by contrast, serves that structural function. And the way that your linkages are formed is, it's a little bit tricky to see in this particular diagram, but your linkages are going to be in these long strands so that you have many strands of your cellulose polymer, and then they are wrapped together into a very long rope-like structure that itself is twisted together. And so you have these long series of twisted ropes sort of woven together. That is going to be something that is very difficult to quickly break down because it's difficult to access all those different sites of those linkages, but it serves a very strong structural function. If you try to break a single thread, that's pretty easy, but if you try to break a whole rope at once, that's a lot more difficult because it has all of those components woven together. So you can see why cellulose would be in that sort of orientation, unlike glycogen and starches, which have this very branched pattern, which makes them really well suited for energy storage. That's all I have on carbohydrates. Pretty quick, hopefully not too terribly painful. The next video will be over lipids, and until then, thank you very much for listening, and keep on learning.